So moving on, we have the DNA, deoxyribonucleic acid, deoxy. That means one oxygen short. It has a double strand, forms a double helix, has a complementary base pairs, longer than RNA. Now you said that DNA is about one strand of DNA is about 7 feet length. Okay, so the three components of the deoxyribonucleic nucleotides, we have the nitrogen base, just we mentioned before. There is ribose sugar. There's one oxygen atom less in ribose. Phosphate group. We talked about this. We talked about it in the previous video. All right, so now look at this. Here is the structure, a full structure of RNA. This is RNA, one strand, one helix, and this is the DNA double helix. Okay, so if you look closely, you will see the phosphate backbone, referred to as backbone. So the helix is called the backbone. And if you look at this picture just it doesn't mean anything. It doesn't actually mean anything in respect to we're not looking at the gene that she carries. We're actually looking at the hair, the arrangement of the hair. So this one strand of hair here is referred to as RNA, while this double strand is referred to DNA. So that's just by the way. So it's not just forget about it. All right, so that is that for DNA and RNA. So we have looking at the structure. We have the linking of nucleotide occur in nucleus during S phase. Now, if we talk about S phase, the previous topic, that's chapter 5. I believe we talked about the mitotic cell division. Now, if you go back there and look at the mitotic cell division, you will see the different stages, the mitosis and the meiosis. Now, talk about mitosis. We have a 5 phase. The first phase is what we refer to as the S phase, the G1 and the G2. So the S phase means synthesis, DNA synthesis. That's what happens in this S phase. So this S phase is where the synthesis of DNA happens. Okay, so that is where the DNA will be referred to as DNA replication. You know that the chromosome inside the cell is 2. They occur in pairs. But before mitosis starts happening, the DNA is going to replicate into 4. Okay, because mitosis will end up producing 2 daughter cells, and each daughter cell must have equal number of chromosomes with the parent, and the parent has 2. So that's why I have to split into 4. Okay, so we call that DNA replication. So that's what happens in the S phase of the cell cycle. So S phase stands for synthesis phase. Okay, so we can refer back to chapter 5 and get to know more about the mitotic cell division. And then also the bond between adjacent monomers after condensation. We have condensation and we have dialysis. Now condensation is where the one that liberates water at the end of the reaction, and this condensation brings about the phosphodiester bond that we mentioned before in the previous part. Now if you look at here now, look at structure of the DNA. But before we talk about this, look at this structure as a whole now. Now if you look here, you see the phosphate group, and we said the phosphate group is negatively charged. That's what makes DNA and RNA a negatively charged molecule. So this is the phosphate group. Now this phosphate group, you see another one here. This is a ribose group. This is the phosphate group. This is the ribose group. Then we refer to them as monomers. Okay, so this is a monomer, this is another monomer. Now when 2 of them combines through the reaction we call condensation reaction, you can see now hydrogen will be, water will be released. Water will be released as a result of this. That's what condensation is all about. So condensation reaction brings about the release of water and then the creation of phosphodiester bond. So when condensation happens, phosphodiester bond will be created, and that's what brings about the phosphate group. Okay, all right. Over here we'll say for the structure, we say DNA has the sugar phosphate backbone. So the backbone comprises of sugar. This is the sugar, the ribose sugar, while this is the phosphate. So we call that sugar phosphate backbone. Okay, so it's formed by alternating sugar and phosphate group. They are linked by phosphodiester bond. Now we've talked about that before. So look at this structure. This structure gives you a full description of what we talk about. So here we have a full structure. You can see here is the phosphodiester bond that exists between the sugar and the phosphate group. And then in between here you see the base. So this one is paired to one ring. This one is paired to two double rings. So here we have the bond that exists between the two nitrogen base is hydrogen bond. So the hydrogen bond holds them together. So the hydrogen bond helps in the holding the two helix together. So it's the work of the hydrogen bond that brings them together. So you can see this is one helix, this is second helix. So this is a DNA because DNA has double helix. And then we have the phosphodiester bond that exists within the phosphate backbone, that's within the ribose and the phosphate, and then the hydrogen bond that exists between the bases, the base pairs. So it exists between the base pairs. Okay, so that's the structure. And here we have talking about the complementary base. Yeah, this occurs the opposite direction. You can see two hydrogen bonds between adenine and thymine. Remember, we said is purine always binds with pyrimidine. Okay, so one purine, one pyrimidine. So in this case now, you can see A is a purine, T is a pyrimidine, and what exists between both of them is a double bond, double hydrogen bond. Now here we have three bond between guanine and cytosine. So guanine again is a purine, while cytosine is a pyrimidine. So what exists with the bond that exists between them is also hydrogen bond, but in this case we have three hydrogen bonds. Now we've already achieved the fact that hydrogen bond is the weakest bond. So in this case now we said when hydrogen bond occur in large number, they are strong together. They are strong together. So it is their number that brings about the firmness of the DNA, because the hydrogen bonds that they are many, so they occur, all of them are there to assist themselves in order to make the whole bond strong. Okay, so hydrogen bond holds the two strands together, easily broken by transcription to RNA or replication. We'll talk about this later in the next part. Now, pairing of bases is precise. We talked about this. So adenine can only combine with thymine or uracil. Same thing with guanine can only combine with cytosine. Okay. Now, two rings plus one ring will give you three rings. Now, distance between strands is the same. So these are what we talked about before. So these are the components. What forms what we call the complementary base pairs. Okay. Now, between complementary pairs, hydrogen bonding, I think base include DNA strand, important for DNA structure. We've talked about this DNA structure, this structure of proteins. We've talked about this in the previous chapter, so you can refer back to the previous chapter to get to know more about the 3D structures. Okay, but that 3D generally brings about, the helix. The helix is the backbone is in a helix shape. So that's because of the 3D structure. Okay, so the different positions where the hydrogen bonds exist that brings about the shape. Okay, so that's what referred to as 3D structure of molecules. Now, many hydrogen bonds give stability. Like I said before, one hydrogen is weak, so but they were cut together to be strong. Okay, so importance of DNA replication, important for DNA replication and transcription, we'll talk about that. So strands can be separated. Hydrogen bonds only form between specific pairs. Fewer mistakes. Hydrogen also can easily reform without chemical reaction. Okay, so that's why hydrogen bond is very important here, because one, they said it can be separated easily, and then it can also be reformed without any chemical means. Looking at the third point, we say two strands of polynucleotide are anti-parallel. Now, meaning that they run the opposite direction. Now, one strand runs from 5' to 3' direction, while the other one runs from 3' to 5'. Now, this is what we mean by when you say 5' to 3'. Now, look at this structure, for example. Okay, before we talk about it, look at this structure here. you look at this structure here, you will notice something. Now, this is a ribose sugar. We know it's the deoxyribose, but let's assume ribose, they're all ribose. Just one is short one of oxygen, the other one is added extra one oxygen. Okay, now each carbon places is what we call prime. We often use this in chemistry. One prime means that the carbon one. Now you're going clockwise from the oxygen here. From this oxygen here, you go clockwise. 1, 2, 3, like that. So going clockwise. So this is first carbon place, which is 1 prime, followed by the second carbon place, which is 2 prime. Then the third carbon place, which is 3 prime, 4 prime, and now if you can see this, the fifth one is outside. Now this is what this is called a pentose sugar. Pent means 5. So the fifth one is on the outside of the ring. Okay, so this is the fifth one. Okay, now if you haven't known this, now let's go to the other structure. Now if you look at this structure, let's look at the membranes are there as well. Now look at where the phosphate groups are attached. Look at where the phosphate groups are attached. Now, as you can see now, this one runs from 5 to, you can see the facing, how it's being faced. Look at how it's being faced. So the last one here is 5. The fifth prime is the one at the end, while the beginning here is a 3 prime. Okay, so the carbon 3 is what is at this end, while carbon 5 is what is at this end. So that's why we have 3 prime to 5 prime. But is in the other way around. The other way is inverted. But if you come to this other side as well, you can see, look at the ribose ring. Look at ribose ring. You can see the inverted form now. If you look at this ribose ring and look at this ribose ring, you will see the difference in how they are positioned. They are inverted. So that's why this one runs from 3 to 5. This one runs from 5 to 3. So that's what we meant by inverted form. Okay, so this one as well, so you have to see the 1 prime, 2 prime, 3 prime, 4 prime. So on the 5th prime you see the phosphate group. So phosphate group is always situated between the 5th prime and the 3rd prime. Okay, so here now the end here is 3 prime, while the end is also 5 prime. So that's why we have 3 to 5. It's not that we are counting from number 1 to 5. No, that's not what we're counting. We're not counting 1, 2, 3, 4, 5. No, it's not that we're counting from beginning to end. No, we are looking at the positions of each, where the prime is facing. So the end here, the prime at the end here is 3 prime that is facing is at the end. That's why I have 3 prime end. While this one is the 5 prime that is facing this end. That's why I have 5 prime end. So the same thing here, 3 prime end, same thing here, 5 prime end. Okay. So that is what this whole thing is all about. It's not that we are counting from 1 to 5, like I said before. We are not counting from 1 to 5. Neither are we looking at the beginning and the end. No, we are looking at the prime that is facing the end, the prime and at the end of the strand. That's what we are counting. Okay. All right. So let's look at this. Look at this exercise, for example. Now I said you only need to be given what one end of the DNA molecule is called to label the rest. So you can our advice, you pause the video and try to label the structure by yourself. Just pause the video for a few seconds and try to label the video by yourself. Okay, I'm going to, I say 2 seconds. I'm going to count 2, 1, 2. All right. So I believe you've looked at this, and I believe you've come up with your answer. Now here is 5 prime. Now for those that couldn't still get what the answer is all about, but here is 5 prime. So if I trace this ring, this helix, see the way, see now that's why it's called helix, and then I trace it to this point. So here becomes our 3 prime. This place becomes our 3 prime, since here is 5 prime. Okay, so if this place is our 3 prime, and we already said that is in inverted form, so here also will be a 3 prime, and then when you twist it again, you come to this point here, it becomes our 5 prime. So you see it now, it's very easy, very simple to understand and to remember. So this is 5 prime. When you trace it down using following the helix, you have 3 prime here. Then if here is 3 prime, that means here it will also be 5 prime. So if you trace it up again following the ring, the helix, then here is 3 prime. So that is the answer. You can see it's very easy. Then lastly, for this path. Now let's look at this question. Look at example questions. I will also advise you pause the video for a minute or two and try to attempt the question. Now look at the question. They've given us 5' to 3', okay, for one helix. Now the question is, given the sequence of DNA above, calculates. First one here said the total number of bases in the length of DNA. The total number of bases in the length. Now they have given us one. Please try and understand the question before you answer. Now you said calculate the total number of bases in the length of DNA. Now for them to mention DNA, we already know that DNA is double helix. DNA is double helix. So anything you are doing, all your calculations should be based on the fact that DNA is a double helix molecule. Okay, they've given us one, so they expected us to find the other helix to make it double helix. Second calculation, say calculate the number of purines and pyrimidines. Now, after you've identified the total number of bases, that's where you now start classifying them based on either their purines or their pyrimidines. Then the last one, then you calculate the number of hydrogen bonds in the DNA molecules. So after getting the purines and the pyrimidines, that will give you a clue on how many bonds you have present. So like I said, pause the video, take 2 minutes and solve. Take 2 minutes and solve. Okay, so look at these are the answer here. So let me explain this answer now. Look at the first one. They've given us one helix, 5' to 3', and they've given us the bases there. We have thymine, 3 adenine, 1 guanine, 3 cytosine, another thymine, and another adenine. Now, the total here, if you count what we have, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So they gave us one helix to have 10 nitrogen base. That means that by the time you add the second helix, which is going to be from 3' to 5', you're going to have a total of 20. So that's why we have 10 times 2, which will give us 20. So the total number of bases in the length of this DNA here is going to be what? 20, because we are dealing with 2 helix. All right, next question. The number of purines and pyrimidines. So calculate the number. So now we're going to identify. Remember, we said purine is pure as gold. So we have adenine. All adenine and all guanine are what? Purine. All others are pyrimidine. So let's identify them. So the highlighted yellow, the highlighted bases here are the purine. So you can see the adenine, you can see the guanine. So all adenine and guanines are purines. Okay, so we highlighted them. The question is the number of purines and pyrimidines. So what total of purine do you have by the time you count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10? So that have 10. So the remaining ones are what? Pyrimidine. Remember, we said purines always bind with pyrimidine in order to balance up everything. Okay, so the last question, calculate the number of hydrogen in the DNA, the number of hydrogen bonds, in the DNA molecule. Now, we've already achieved the fact that 2 bonds is what holds adenine and thymine. So 2 bonds, while 3 bonds what holds the guanine and cytosine. Okay, so with that, since we have 2 bonds, so how many adenine and thymine do you have? 1, 2, 3, 4, 5, 6. So because there are 2 bonds, I will have 2 times 6. Because each of them has 2 bonds, so I have 2 times 6. Okay. Then for guanine and cytosine, how many guanine do we have? We have 1, 2, 3, 4. 4. So if you have 4 guanine, we're definitely going to have 4 cytosine. So we'll go 3 bond times total number, which is 4, which will now give us, by the time add the 2 of them together to give you what? 24. Okay, I believe 2 times 6 give you 12, 3 times 4 give you 12. So everything is going to give you 24 hydrogen bonds. Okay, so I believe you understand that. If you don't, still you can ask me, you know where to reach me. You can ask me questions more on this. You can drop it in the comment section. That's it. Okay, so I think we've come to the end of this part. So thank you very much and see you in the next part. Thank you. Bye.